My name's Kevin Mitchell, and I'm from the Department of Genetics here, and I'm going to talk to you about genetics, or what the science is about, and what the course is about here, and if you come as a student here, what kinds of things you'll be learning. So the title of the talk is rather grand. It's called The Secret of Life, but the reason is that's exactly what genetics is about. Genetics is about the difference between this frog and this pile of dirt. Okay? So they're made of the same stuff. You get carbon and hydrogen and oxygen and nitrogen, some other atoms and elements and stuff in there. Okay? But the difference between this and this is organized. That's organized matter. Okay? That's the difference. And now you might say lots of things are organized matter. Your refrigerator is organized matter. Right? Your iPhone is organized. Okay? The difference here is this thing organizes itself. Okay? This thing has within it the code to make another frog. Okay? That's what genetics is about. It has the information inside it to both keep itself running, to keep itself ordered, to stop itself becoming a pile of dirt, which takes a lot of work, and also to make another frog. Okay? And that secret, that is obviously the DNA inside it. That's where the information is that each organism has inside it, the instructions to make that organism, the instructions to make the cells, to make all the bits of the cells in the right places, and to keep everything working. So inside each cell, there are the chromosomes which contain all of the DNA, which has this particular sequence, and the sequence along the DNA codes for proteins. And the proteins are these little molecular machines or little robots in the cell that do all the jobs inside each cell. Okay? And different cells make different proteins. So in humans, we've got about 30,000 different genes that make about 30,000 different proteins, and each of those is expressed in different cells to control all the operations of the cell. Now, the difference between this organism and this one is that this one has frog DNA and this one has grasshopper DNA. Okay? And this one has some kind of plant DNA. This is some other kind of plant DNA. Okay? So the genetics is about not just understanding how living things operate and how DNA works, but understanding the differences between them. So what is the difference between the frog DNA and the grasshopper DNA? When you start with a single cell, a little grasshopper egg, little frog egg, what is it about them that makes one of them develop into a frog and the other into a grasshopper? All of that information is there in the DNA, and geneticists try to figure out how that works. What is it, that sequence of A's and T's and C's and G's? Where's the magic in there that actually encodes that information that makes one organism different from another? And obviously you can look at that not just from one species to another, but within a species you can say, what's the difference between individuals that makes them different from each other? And that's obviously something that geneticists are very interested in too, especially in humans. So you can look at some particular region of the DNA sequence of the genome. This person might have a T where everybody else has a C. Okay? And that might make some difference to that person. Maybe it makes them have red hair, or be taller, or have cystic fibrosis, or some trait or some disorder. Those are the ones that we are interested in tracking down to see if we can understand how differences in the DNA contribute to differences in what we call the phenotype, or how everybody is, all the traits of a person. And they can do that because sometimes those differences can, if say this is a sequence that makes a normal protein and you get a mutation here, that can make a damaged protein where it doesn't work, and that can cause a disorder, or it can cause just a difference that's not actually a disease. But that's the nature of how DNA changes affect traits, and that's why DNA is the molecule of heredity. That's how traits are passed on, because if one person has this, then their offspring may inherit that as well. And so that's why people resemble their parents. So what we're interested in, here we've been studying genetics at Trinity for over 50 years. The department was founded in 1958. Our philosophy really is for research-led teaching. And what I mean by that is that we don't, in school you guys get taught science like it's a kind of like a body of facts. Like it's a static, dead thing. You just have to learn A to Z and you're done. Once you get to college, it's not like that. The point of the way we teach, at least, is to tell you how to become a researcher, to show you how to think like a scientist. So we're always at the edge of any subject, the edge of what we know. We say, okay, this is what we know so far, but this is what we don't know, and this is how we would find out. Okay, or this is how people found out in this paper that was published last week, or this is how the research that we're doing here in the department lets us find out. So the point of it at that stage is to make you able to think like scientists and think like geneticists and understand how research works and understand how science develops, not just know the facts, because the facts will be, well, there'll be new facts 5 years from now. So the importance is an education that's really broader than just the information. We have small classes, so there's about 30 students in our third and fourth years, and we have about 15 faculty, so a very good faculty-student ratio. A very collegiate atmosphere. We're on first-name basis with all of the students in the department, and the Genetics Institute is ranked, or has been ranked at least first in the world in molecular genetics and genomics. So when I say we do research-led teaching, that means really it's top-notch, international quality research. So there's two entry streams to the course. They're sort of parallel. One is to the entry to the general science course, which is TR071. And after first year and second year, in which you get a sort of general grounding in various subjects, then you can specialize in third and fourth year in genetics. Now the other stream, it runs parallel to that. It's a direct entry course from the CAO. It's called Human Genetics. And when you come into human genetics, you're basically on this track already. You end up in human genetics in third and fourth year. And the main difference is as you come through here, there's more particular tutorials and things as you're coming through. And also then at the end, when you're doing a project or doing literature reviews and things like that, it'll all be focused around human genetics. There'll be, including maybe research placements in hospitals like labs up in St. James's or the clinical genetics labs in Crumlin and so on. So overall, they're similar, but there's more of a focus on the human side here, and it's a direct entry course. So as I said, there's 2 years general science. There's a specialization in genetics in third and fourth year. We have a very good student-staff ratio. Our philosophy here in third year is to give you really a grounding of all of the areas of genetics. So genetics is fundamental actually to every aspect of biology. So if you're interested in neuroscience or immunology or evolution or plant science, you can approach all of those topics using genetics as an experimental method, and that's what we do. And so what we want to do is in third year is give you that grounding so you're familiar with all of those areas of genetics. And then in fourth year, you can choose to specialize. If you're more interested in evolution or medical genetics, you can focus your research there. You can focus the kind of lecture classes that you're doing and reviews and so on. The final year, we have advanced classes, and what that means is that each of us, all of the faculty, are teaching classes really in our specialized subjects right at the forefront of research as it's happening. So, it wouldn't be unusual for someone to find a paper that's just been published that morning or the day before and bring it in and talk about it with the students in the lecture. So you're fully up to speed with the state of the art in whatever the field is. And the idea is not so much that, again, it matters that much what the field is. The point is that you learn how research works and that you get a really up-to-date kind of grounding in that. In final year 2, we're looking for a lot more independent research and work from you guys. So you do an independent literature review on some particular topic. You do a senior thesis research project in one of our labs, which lasts for about 12 weeks. And that's a chance to get into the lab to see actually what scientists do all day long, to do some hands-on research to see, okay, maybe it doesn't always work first time the way you think it should. That's just the nature of experiments. But generally, students get a great experience out of that. So this is just an overview of some of the topics of genetics that are covered both in the lectures and also in the research. It goes from everything from molecular and cell biology. So this is looking down into cells saying, okay, what is the gene that makes this protein? What does that protein actually do in a cell? Maybe it controls DNA replication, maybe it controls cell death or how much cells replicate, something like that. And you can get down to that level and really experiment with the cells and see what's happening. You can get up to higher levels with looking at the effects of genes in organisms. how they develop, for example, and especially things like medical genetics, so which mutations in particular genes can cause diseases, and then all the way up to things like tracking populations, population flow using genetics in the same way that you can track a paternity test, except you do that over many, many generations and over whole populations, and even all the way back to whole species. So you can do that, the same logic, use DNA to see who's related to who, but over millions and millions of years of evolution. So let me give you a few examples just from the research that's gone on in the department over the years that illustrate some of these different topics. So one of them is research that Professor Jane Farrer and her colleagues have been carrying on for about 30 years now, actually, which started with looking at families that have an inherited eye disorder called retinitis pigmentosa. And what happens in that disorder is that these cells in your retina, which are the photoreceptors, the ones that actually detect light, degenerate over time. The reason is one of their genes is mutated. It makes a protein that actually is toxic to the cells. And over time, as that protein, the toxic protein builds up, the cells actually die. Okay? Now, the effect of that is that the person will lose their vision, and they lose it initially around their peripheral vision, but eventually they'll go blind, usually by mid-40s. And there's no cure for that. And so that just happens, and it gets transmitted in families like this. This is one very large family pedigree where females are circles like this and squares are males, and the ones that are filled in black are all the people in this pedigree who were affected with this disease, who have all gone blind. Okay? So what these researchers did was track that disease and see where in the genome, when the DNA is passed along, which is the bit that all of these people have in common. And by doing that, they were able to track exactly where this mutation was, find the gene that was mutated. It turns out to be mutated in many other people in the world with this condition as well. And then figure out something about the biology of it and understand that actually the reason for those cells dying is because that protein, because you have a mutation in the DNA, changes that sequence. And when that makes a protein, then the protein is altered and it's toxic. Now that work took well over 15 years. And what they've done since is gone on to make a mouse model. So what they want to do is not just understand the cause of the disease, they want to use that information to be able to treat the disease. And the way that they're doing that is to, first of all, make a mouse model. So they can make a mouse that expresses that protein, and their retina degenerates and they go blind. So it's a system that you can use to develop treatments. And the treatment that they're using is actually another genetic trick to chop up the mutant DNA. Okay? So what that will do is stop the gene from making that toxic protein. And then they can replace it with a normal protein. And that's showing really great promise in these animals. It can restore the sight in these animals. It's now in preclinical trials in monkeys. And there are some other conditions like this that other people in the world have now done injections of virus into the eyes of patients where they can do a gene therapy replacement like that and correct a genetic disease directly in the cells of those patients. So that's one of the goals. It sort of illustrates the idea of going from really early human genetics through animal models and so on. This is another one which is an unexpected sort of a story. Remember I said you can use DNA variations to track populations and see where they came from. And for example, in the Irish population, you can see that people came from, there's some Viking DNA, there's some Celtic DNA, there's some Norman invasion DNA in some people and so on. You can track all of that. Well, you can do the same thing for species like cattle, and there was a group led by Dan Bradley here who was interested in that. And what they wanted to see was a really basic research question. Were cattle domesticated once in the world, and then that idea sort of spread everywhere else, or were they domesticated twice independently? And it turns out that they could see that actually they were domesticated twice, once in Africa and a different species here in the Middle East. So that was interesting. It's sort of an academic question. But what came out of that was that it was actually a business opportunity, because what they realized was if we can trace cattle back, hundreds and hundreds of generations, then actually we should be able to trace your steak back to the farm that it came from. And that became important when the mad cow disease scare was out, and a foot and mouth disease and other things like that. And it's important now just for consumers who want to know if their cow is from an organic farm or not. And so this DNA traceback technology, which you might see on your steaks next time you're doing your grocery shopping, is is done by this company called Identigen, which came out of the genetics department here and is a very successful company. Now, they've gone on to actually look at, as I said, the genetics of the Irish population, but not just early. They can actually get ancient DNA. So you can get DNA. DNA is one of the most stable molecules around, actually. That's why it's good to have as a code for life. But it means that you can get DNA out of ancient bones, ancient dinosaurs and Neanderthals and things like that, including the bones of people who were ancient Celtic people who may have been buried under things like this. So what this guy is doing is drilling out the sample from the skull of someone like that to get DNA to see where those people came from and to basically get a look back in history. Okay, I'm just going to skip through a couple other things just to give you an idea of the breadth of what goes on. There's people working on bacterial genetics trying to understand, in this case, how bacteria make their cell wall. So the outer wall of a bacterium made of different proteins. It takes enzymes to make those things. And that's really important because a lot of antibiotics work by attacking the cell wall of a bacterium. And unfortunately, antibiotics have been so prescribed and overprescribed now that they're losing their efficacy because the bacteria are evolving new ways to become resistant to them, which means we urgently need to know more about the biology of these pathways in order to design new antibiotics. Otherwise, we're going to hit a time point when infections become really, really serious again. There's people working on cell death, so understanding when a cell decides to kill itself, which may seem an odd thing to do, but actually there's programs in the genome that are designed for a little self-destruct process, especially if a cell gets too many mutations in it. And that's actually a protective thing against cancer. So if cells get too many mutations, they can become cancerous and overgrow. And one of the ways that they do that is by short-circuiting this self-destruct mechanism. So understanding, again, the processes by which that happens inside a cell is really important. We may be able to trigger that in cancerous cells to get them to kill themselves. There's people working on genetics of flower development, which is important for lots of agricultural reasons. People working on neurogenetics, so this is my own area, understanding how the brain develops, how different parts of it get connected to each other during development. This is a mouse brain. This is my brain, actually, which is awesome. And this is a little neuron that is growing, and it's trying to, it sends out a connection. It's trying to find a partner, basically. It's looking to see who it should connect to. And there are genes that control that. They're all in the genome of the fertilized egg. So all of the information to make a baby is in there. And what we want to do is figure that out, and especially figure out in the brain how you make all these gorgeous connections, which is really complicated process. And if you get down to the cellular level, then you can see these little synaptic connections between neurons where they pass messages to each other. And those, again, are really highly specified during development, which cells connect with which in order to get all the information going to the right places. One of the reasons that's interesting, too, not just as a scientific question, is that defects in the proteins that control those processes, so mutations in those genes, some of which are shown here, these little schematics are little different proteins that are connecting different cells. Mutations in those genes can cause things like autism and epilepsy and intellectual disability and schizophrenia. So we're trying to figure out the biology of how these things happen and how mutations in these genes can lead to conditions like that. And actually, there's been a massive leap forward in that in the last few years, where, for example, for autism, even 5 years ago, we could explain maybe 3 or 4% of cases of autism down to a particular mutation in a particular gene. Now we can explain maybe 30%, and that's growing all the time. So it's a really, really rapid, exciting time in the field where there's lots of progress being made for disorders that for decades have been a mystery, actually. And that's really a general thing because there's an enormous explosion of information and opportunity here in personal genomics because of the advances in technology and DNA sequencing. So we can sequence genomes just much more cheaply than we could before. So in fact, if you look at the cost of sequencing a genome, the original human genome, when it was sequenced, actually cost, this is even an underestimate, it cost $3 billion and it took 10 years and hundreds of people to do it. You can sequence a human genome now in less than a day for $1,000. So that changes things. That changes how we're going to do medicine. Okay, when you come into the doctor in the future and you have some ailment and they don't know what's wrong with you, they'll run a bunch of tests, but they'll sequence your genome. And someone's going to have to tell them what that means. And that means there's going to be a huge market for people with skills as geneticists and that sort of information. And you can see that Angelina Jolie is up here because she was in the news there about a year ago where she had inherited a breast cancer mutation. She knew there were people in her family who had breast cancer, I think her mother and her aunt. And she got her DNA sequence, found that she had that mutation. And unfortunately, there's not really a treatment, but there's a preventive thing, which is a double mastectomy, which is what she had. And she was very open about that and actually spread a lot of awareness of that and the importance of getting that test if you have familial breast cancer. But it's an illustration of the fact that medicine is going to become much more personalized. It's not just going to be throw the same drugs at everybody. You're going to come in, you're going to get some genomic characterization and say, okay, this is why you have autism, or this is the type of cancer that you have. It's going to respond to that drug and not to this drug. And in fact, if you look at the original DNA sequencers, there were these warehouses much bigger than this room with banks and banks of enormous sequencing machines and hundreds of people working on them. Nowadays, this is what a DNA sequencer looks like. It fits in the palm of your hand. It has a little USB connection. It sticks right into your laptop, and you put some blood in there, and your genome sequence comes out directly into your computer. So this is not the future. This isn't science fiction. This is happening right now. It's the future of medicine because it really will change how we do everything. And not just medicine, it'll change all kinds of things in biology generally. Okay, just one last note on what our alumni do. About 2/3 of them go on to do a higher degree in master's or a PhD of some sort, and many end up doing research. A lot of them, for example, we have professors all over the world, including in Columbia and Memorial Sloan Kettering, Cornell in New York. People have gone into biotechnology. These companies have come out of the department. And then, people go into all kinds of things where they're using the education that they get, maybe sort of directly, maybe not, in things like medicine and law. And a number of people now gone into science journalism, teaching, various government agencies and so on. So there's a lot of opportunities outside just becoming a geneticist where having that education is useful just for general critical thinking, but also the knowledge that it gives you. Thank you.